Okie dokie.
Hello and welcome back to Real Seekers. I'm your host, Dale, the Real Seeker. And today I have an extra special treat. It's something that I have been waiting for at least a month and a half now, because I know everyone's familiar with Kevin, my co-host, Kevin Nontradicath, or Kevin O'Connor. Hey, Kevin.
Hey Dale, thanks for having me on.
No worries, no worries. Just very briefly, what have you been up to since the last time you were on the show?
Oh man, I guess mostly non-YouTube work stuff. But I did get a lot closer to Lanciano, Italy than I normally am. And for those of you who don't know, Lanciano is the first Eucharistic miracle in Dr. Serafini's book anyway. And that's just because I took a work trip to Italy. So it had nothing to do with Lanciano. I was in Milan, not Lanciano, but I was a lot closer than I normally am. So that's all.
Awesome, awesome. And obviously we have the main guest of honor, Dr. Kelly Kearse, who's been on the show. Hey there, Kelly. How are you?
Hey, how good? Good. How are you?
I'm very good. Yeah. Great to have you back. I
Yeah,
usually bring
thank
you
you.
on for topics related to the shroud and the
Right, right.
but you have written a recent paper on the issue of Eucharistic miracles.
Mhm.
So I wanted to bring you on as an expert on that front. But just before we get into the topic, do you just want to let the audience know what you've been up to since you were last on the show?
So pretty much the same thing. So I'm a teacher. I teach high school science. I do that, and then I've also been working on shroud things, shroud slash forensics type things since the last show. I have several irons in the fire on that.
Awesome. Awesome. All right, cool. So, yeah, with that said, let's get straight into the topic of Eucharistic miracles. And Kelly, I just want to kind of get a general notion from you. Like, what kind of interested you in the topic of Eucharistic miracles and kind of influenced you to actually study this?
Okay, so I'm a Catholic. I'm a convert. I converted as an adult. Years ago. I myself have been a Eucharistic minister for over 20 years. Um, what got me into it is the number one question that I would be asked through if I gave a talk in the parish or something on the shroud, or somebody asked me about the shroud, or a lot of times I get emails just kind of out of the blue questions. The number one question that was asked, well, what about these Eucharistic miracles that type is AB? Because the shroud type is AB as AB. Can there be some sort of connection between those? So it was just a question that just kept popping up and popping up and popping up. So after, I don't know how many of them, okay, I'm going to look into that and just sort of see. And I must say, now I didn't really know that much about them, to be honest with you. What I thought happened with them is that the way it would go with these bleeding hosts and things like that is that that would happen at the moment of consecration when the priest was praying. I thought, boom, that's when you would see the reddish blood. But so the first miracle I read about, it was that a person had found a discarded host in the back of the church, and normally a priest would consume a host that's discarded, but these are very dusty and dirty. So the protocol there is to place it into water for a special disposal. It wasn't until after placing it in water 7 to 10 days later that this reddish bleeding started to occur. And so when I read the instance on the first one, it was kind of a flag. It was sort of like, wow. So first it's put into water and then you see it. And then I read about another one, same thing. Read about another one, same kind of thing. And I just wondered, why does Jesus have to be in water for a week in order to see that that's when you have the blood start to appear? It could be just something that's growing there. Now, there is one miracle, Tixtila, that's reported. A nun is distributing communion. I'm pretty sure that's the right one. She was distributing communion and noticed that there was a host that had reddish areas on it. But you can imagine that moisture had gotten in there through humidity or maybe just sweat from someone's fingers or something like that. And it's closed up, kept in a dark container. So you can imagine that it's a similar sort of scenario. So there was a very similar kind of pattern to this, which really surprised me. Again, I thought it was kind of instantaneous at the moment of consecration. So with that in mind, I thought, well, what happens if you take a non-consecrated wafer and just run it through the same kind of conditions? And I looked around and no one had ever done that before. To me, that's kind of an essential experiment to do. So that's what led me down the path. And I did this, it was kind of a summer project together with a student, Frank Lagai, the second part, the one on the non-consecrated wafers. So, but it was the AB that with the shroud, that was really the gateway that got me into doing it, just because I was asked about it so many times.
Yeah, awesome. And just for the audience, I'll just show the audience very quickly, share my screen for a second, and just show them that this is one of the papers that Kelly's published, the scientific analysis of Eucharistic miracles, importance of a standardization and evaluation. So just want to show that for people to check out. But yeah, just before we start getting into some of the specific cases, Kevin, I just want to turn it to you as the co-host. Do you have any sort of general questions for Kelly?
So I have specific questions about the studies, but I think those are probably better served for later in the call.
Mhm.
Um, but if I can just go on record and say that I am super grateful for Kelly's work here. I have been I feel like screaming into like a void about how like there's something wrong here, and it should worry Catholics that people seem to be using these Eucharistic miracles as like real cornerstones when the science simply isn't as robust as they might think that it is. Uh, and so these two papers from Dr. Kearse have just made me feel sane again. So I'm just super appreciative. I'm really looking forward to the slides, and then I do have a couple of specific questions that I hope aren't so specific that we lose our audience, but they're on my mind, so I'll ask them later.
They like the details on my channel, so for
They
sure.
sure do.
I do have one last general follow-up for you, Kelly,
Sure.
because this is another interest of Kevin, and I'm also having Massimo Paris, who's into the shroud, come on my show. But Marian apparition. So you are a Catholic. What's
Right.
your general take on Marian apparition? So,
I haven't really read about those in detail. So I believe that a Marian apparition, Eucharistic miracle, I believe all of that could happen as a Catholic. I have zero problem with something miraculous like that taking place. So the purpose of the studies would not to try to, I'm going to show you why these aren't true kind of thing, but just to explore them and scientifically, objectively look at them. And at the end to provide kind of a checklist or blueprint about, I think there needs to be some systematic type scientific protocol that is gone through when these things occur. And it's done more in a typical scientific context with the goal to publish it in a peer reviewed journal to make it very transparent. So that was really the goal of it, not to say that, well, this is why these aren't true, but just to explore them. I think it certainly could happen. Marian apparition, Fatima, a lot of people saw this. I mean, I have zero problem believing in it. My faith would be very strong that that kind of thing could occur. But as far as detail and stuff, honestly haven't looked into that much. But you have something that's seen by a lot of people as it occurred, which would kind of put it in a little bit of a different, category there.
fair enough. Yeah, kind of like the group appearances versus single or
Right,
something.
right,
So.
right.
Okay. All right. Cool. So I want to start getting into specific cases, and I think we should take it case by case. So
Okay.
obviously, as Kevin hinted at, the oldest is the Lanciano claim, dating back to the 7th century there. So yeah, Kelly, I want to turn it to you and present your case. What were your findings about the Lanciano? First of all, what is it, and then what were your findings on it?
Okay. Do you want me to do that first? Do you want me to do the AB first, which kind of is all of them across the board? Or you want me to start with Lanciano? I can do either one you like.
Okay. So if Kevin.
So I actually think that Kelly's plan here is a pretty good one, only because the same sets of problems tend to impact each one. So having a background on, for instance, how AB blood typing, like how the testing works, I think would probably be useful. That's just my 2 cents, though.
Yeah, sure. That's
So,
good.
Okay, so why don't we'll roll with that one, and then we'll take it right into Lanciano after that. Is that cool? Okay, great.
Yeah.
So let's see. I'm going to share and go there. And do you guys see that on the screen? You see the pie chart?
Yep.
Yep, I can see it.
And is the stop share thing, is that, do you guys see that? Is that in the way or is
No,
it okay?
you just
Nope.
see that on your end. We don't see
I
that.
see. Okay, let me just make, okay, great. So here we have, this is just a general distribution of blood type. This is going to vary a little bit depending on geographical location and population. But in general, most people are type O, and in a pretty close second place would be type A. A pretty distant third, B, typically, again, some variation with population around 8%. AB is the rarest, usually around 3 to 5%. Again, some variation depending upon population. So you have a blood type, AB, that's the rarest of all of them. And we have reported for the shroud of Turin, the Sudarium of Oviedo, a face cloth that's believed to have been placed on the face of Jesus after death, and then the tunic of Argentuli, a robe thought to have been worn on the way to Calvary. All of these test as AB. And we have various Eucharistic miracles, all of them which have been tested to date. I believe it's up to 5 or so now, but all of them that have been tested to date type is AB. So it's been reported for just 5 of these kind of objects that it's statistical improbability for this to happen merely by chance. The odds are greater than 3.2 million to 1. If you factor in some of the others, it can get even much higher. In Catholic Answers in 2023, they wrote that this is mathematical proof of the real presence, the fact that the AB is shared, proof that God is real, and proof that the Lord has AB blood. So as far as what A and B, what that really means, if you look at the structure of the molecules, these are composed of various types of sugars or carbohydrates. So each of these little shapes represent a particular kind of sugar when it has a different color. And if you look at this middle part, that's exactly the same in A and B. But if you look at this one on the side, it's a different sugar that's present on A versus B. So that's what the difference is between A and B. It's just a single carbohydrate or single sugar difference. What's overlooked a lot of times, I think, is that many bacteria also express identical A and B molecules that you find on human red blood cells. They're structurally identical, exactly the same thing. So bacterial contamination, it's been reported for many of these objects. They're certainly not sterile by any means. And when people look for bacteria on there, they find that a lot of them are pretty heavily contaminated. So I think that's a plausible explanation that should be considered when all of these things type is AB. The way these tests are done, that have been done for all of these objects, including the miracles, that are serological methods, which just means that you use antibodies to recognize the A and B. And these antibodies, they cannot distinguish between an A and B on bacteria versus an A and B on human red blood cells. To the antibody, these look identical. The way the shroud studies were done, this was done by Baima Bologna. He's an Italian scientist. This has never been published in a peer-reviewed journal. It's only reported in like specialty journals and books. But he took blood-stained fibers of the shroud and found that they were positive for AB. When he looked at white fibers without any blood in an adjacent area, they were negative for AB. Now, this is a very good type of control to run, but you could always make the argument, well, those bacteria are going to be localized where the food source is. So you're naturally going to find them concentrated in the blood area. Sudarium of Oviedo, similar kind of studies using antibodies. Now here the blood stains type for AB, although the B was a little bit higher than A. There is something called acquired B syndrome or acquired B blood type, where bacteria have an enzyme that can convert A to B. In the Sudarium, in the off areas, there was also an anti-B signal there, which is a little bit of a concern. Way to sort this out to see, well, is it is the AB signal truly coming from human red blood cells, or is this coming from bacteria? You could turn to something called glycosyl transferase enzyme. It's a very brutal word, but what the glycosyl part means is sugar. So these are the enzymes that add these sugar chains to either human red blood cells or to bacteria. We're just going to call these GT for short. So they're not involving the structures themselves, like what an antibody would recognize, but these would be the enzymes that would add that structure. And we're going to look at this at the DNA level. So there's the GT that is responsible for the A blood type in human. That has been described, very well characterized, as has the GT for B in human that transfers that structure. So you can tell those two apart very, very easily in humans. This is usually how blood typing is done in modern day uh, type studies, is at the DNA level. But the GTA for bacteria has also been isolated, and it's different than the one that's in humans. So there's a discrimination there at the DNA level. You could tell whether that the human GTA was present or the bacterial. Same thing for the GTB that you find in bacteria. So looking at it at the DNA level, that's one way that you could actually discriminate between this type of signal. And so just to kind of recap that, the serological methods, this is what's been used, antibodies, in all the studies to date. They cannot distinguish between bacterial AB and human AB. But if you looked at this at the DNA level, that could potentially provide an avenue to distinguish, well, is this really a bacterial type of AB signal that you're seeing, or more toward a human AB signal that you're seeing? And I'd also just like to make the point that shared AB among these different miracles or objects, that alone, it's not sufficient to demonstrate the claims of a single origin, which is really what you have to do to suggest that these come from a single source. Now, you could have two people that are AB blood type, but of course they could be different individuals. To do this, you'd have to go further than AB. So you'd have to use markers that are unique among individuals. So one possibility you could do is look at nuclear DNA, particularly something called STRs. They're about 3% of the total human DNA, and they're unique among individuals. This is what they use when they use DNA profiling in criminal cases and things like that. Mitochondrial DNA, a similar kind of theme. There's hypervariable regions that are unique among individuals. And in the paper, I also suggest, well, you could use HLA transplantation antigens. HLA transplantation antigens, this is what is looked at if someone goes in for a transplant and they try to match to see, well, they're going to be a good recipient or good donor, because these vary among individuals. What the HLA, the advantage of that, it has a unique opportunity to look at this both at the DNA level as well as the protein level. With DNA, contamination can always be a kind of concern, and they've run into some problems with the DNA and analysis of some of these miracles, which we'll talk about. They show these very pristine kind of textbook cardiac tissues as example, ideal for testing at the protein level with the HLA. Before DNA technology really took off, this is how they did tissue typing, was using antibodies at the protein level. At the protein level, the contamination is a complete moot point. It's not an issue. So to kind of sum that part up, with the HLA, you could distinguish this using antibodies. You could also distinguish it at the DNA level, but you really have to take it past the AB to make any kind of claim. And the fact that you find AB on bacteria, identical structures, which the antibodies cannot distinguish, I think that has to be taken into account before you can start putting up odds, 3.2 million one or greater, or things like that. To me, it's a very plausible, reasonable alternative explanation. So I'll just, I've got the Lanciano keyed up here, but I'll just pause if you want to ask anything else about that, or. It's not really surprising that these things would turn up as AB. I think that's kind of what you would expect with these type of older relics, and the same with the miracles as well.
Can I ask a question that
Sure.
I know the answer to, but I think somebody in the audience might want to ask this question?
Okay.
Um, so somebody in the audience may be a faithful Catholic and may have read Dr. Serafini's book, A Cardiologist Examines Jesus. And in that book, Dr. Serafini quotes from Dr. Linoli's 1970, I forget
71
the
or
year,
70, I think.
71,
Yeah.
yeah, report in which Dr. Linoli says, referring to the AB blood group here, the delicate absorption elution test allowed it to be objectively concluded with full certainty that both the blood and the flesh of the Eucharistic miracle of Lanciano belong to the same AB blood group. Kelly, you said something about bacteria and you said something about contamination.
Right.
Dr. Linoli sounds pretty convinced here. What help me kind of piece this together. What is this disconnect? Can we be fully certain?
So, no, I don't think so. So I think you run into the same problem. You don't know that whether the AB signal is coming from bacteria or from human when you're using those type of antibody tests. I'll talk about this a little bit in a minute, but I think you could also have some non-specificity of the antibody binding. That wasn't shown in the paper. It wasn't addressed. So I think that's another potential problem with that. Those I don't remember exactly what the words, but they sounded pretty strong, like unquestionably or object,
Mm-hmm.
shows. No, I don't think you can really go there. I think you have to consider the bacteria first and foremost, because the antibody's not going to be able to tell the difference. And I think that type of absorption elution test, it's based on when you see those red blood cells that are agglutinating in the panels, those are fresh test red blood cells. It's based on what antibodies stuck to the original sample in the first place. You can have non-specific sticking in those kind of tests. You have to include those kind of controls for the non-specific binding anyway in any kind of ordinary scientific test. But in an extraordinary claim, I think you really have to go above and beyond doing that. Now, I think Dr. Linoli, a lot of respect and commend him for doing these experiments and publish them, especially publishing them in a journal. I think that's the route to go. But he was also kind of somewhat limited by the technology that was available back in the early 70s. But no, I don't. I think you run into those inherent problems.
And so this is a legitimate question now. I actually don't know the answer to this one. Would a doctor in 1971, such as Dr. Linoli, have known that bacteria or did we know in
So
71 that bacteria give off both A and B?
potentially. So Dr. Springer in the 60s uh, would describe that A and B are expressed on bacteria, not at the
Hmm.
genetic level, but serologically. So there's a list of bacteria that express these. It's referenced in the paper. They're not rare type bacteria. It's very everyday common bacteria, E. coli, Heliobacter, etc. So I may or may not have been aware. I would say someone that's an immunologist with an immunology background, I would say they would have been aware and could have taken that into consideration.
Cool. Thank you.
So possible. It sort of depends on the background or the specialty, I would say.
Yeah,
And
and I was just trying to give Dr. Linoli the benefit of the doubt here because 71, I know it's not that long ago in terms of all of history, but in terms
Right.
of medical advancement, 71 is a long time ago.
Exactly.
It's
Exactly.
not that
Yes.
long ago that, um uh, just recently I did 23andMe. I got my whole genome sequenced.
Okay.
I couldn't have done that 10 years ago even really.
Sure.
It would have
Sure.
been way more expensive. So,
Sure.
yeah, I just wanted to give him the benefit of the doubt.
So I think it's a reasonable thing for him to conclude, but I also think that if an immunologist might have looked at it, I would think they would have raised the point even back then
Mm,
as a possibility.
okay.
Okay. And just one quick follow-up general question on this before we get into the Lanciano. But so I'm interested, what about other relics with, have there been other similar testing for AB on other ancient relics with blood on it? And how did those come out? Do we get
Are you
this
talking about like religious relics?
uh,
So
just
we have the shroud, we have the siderium, we have the tunic. Those are all AB, for those that have been tested.
Mhm. And but even outside of religious relics, like
Outside of that, so you can find, there's you can find some studies. Usually, there's a little bit higher. It depends on how the tests were done. There's called forward typing and reverse typing, but most of these are done by forward typing. So you'll typically see a higher incidence of AB that occurs in these older ones. Now, it's not like 100% or 99%. It just depends on the particular environmental condition, et cetera. You see some that might be boosted up for B because maybe a shift from A to B from a bacterial conversion. It's with older samples, you kind of always run into this problem where you're really not sure, especially with these type of serological tests.
Gotcha. Okay. And I do have a question. This one came from a Shroud skeptic, actually. When I was on S.J. Thomason's channel, they were saying something like, well, it can't be AB blood because the AB blood group didn't exist until the 1600s or so. How have you heard of this kind of skeptical claim? And
I've heard of that kind of thing. Now, I don't really know that much about it. I would think that it would be around at that time, 1600s, like 1600s. I would think, but I'm not the expert on that by any means. So I think the general understanding is that A was the first a very, very long time ago, and then B, and then O, but time frames, etc. I've written people that do that type of thing, and what's the oldest person that's ever been described or remains that is AB, like genetically, that you can find genetically is AB, and the answer is kind of not a lot of work has really been done for that. So whenever they find like a Neanderthal, a group of skeletons or something like that sequence, I'll usually write that person, hey, if it's not in the paper, what's the blood type that you found? And typically O is the response that comes back. But I'm not an expert as far as the time when that frankly there hasn't been a lot of work to really piece that together. It's good questions, very good question.
go ahead, Kevin. I see you want to say
Just
something.
yeah, wouldn't blood type O essentially be the same thing as saying, look, we didn't find any A or B antigens or whatever? Like, um,
So I'm sorry, just could you ask the question again?
so like if somebody's trying to say that a caveman had blood type O, isn't that essentially just the same thing as saying we don't know? Because O is essentially the no
No, no,
result.
because there's a you could test that at the genetic level.
Oh,
Yeah,
okay.
yeah,
We know which genes express the A and the
yes,
B.
very
Okay,
well, very well
okay,
worked out. Yes,
okay.
yes, yes.
Thank you.
You bet.
Awesome. So yeah, Kelly, over to you to kind of continue with our first case study with the Lanciano
Okay.
miracle.
So this is the one that Lanciano is usually when you hear about these. This is usually kind of the one that's first and foremost described. The oldest one that was published in a journal, as we talked about, with the Dr. Glenoli. So he described the blood consistency as uniformly hard. Had to take a scalpel, really sharp instrument to try to cut that off. And in some cases, he just used the blood rinse from it. He described it as being a fungal contamination was very evident, and a chemical test for hemoglobin, which is a signature blood molecule, that was negative. So he did another test called TLC. This is a thin layer chromatography analysis. So just talk real quickly about that. This is a TLC plate, which is usually made of a kind of gel like silica or something like that. And there's these wells in where you load the sample. And you're going to place this whole thing inside of this large chamber. And then you're going to add some solvent liquid at the bottom. And what the liquid will do, it's going to be taken up by the gel. It's going to go into where the sample is, and it's going to cause the sample to migrate up the plate. So a typical result might look something like this. So the sample, you don't really know what that is, but you just look at to where it migrates based on its affinity for the solvent and the plate here, and it's going to stop at a certain point. Control one, you know exactly what this substance is, so you can see where it goes to. Control two, you know exactly what this substance is, you see where it goes to. So ours seem to have a similar migration. It stops around the same point of this. So we probably conclude, well, we have something that's the same thing here as uh, control two. So here's the actual result from the paper. It's a little bit faint to see, but this is done in triplicate. And here you have things that migrate around where blood migrates. So it certainly is I would say, does it show that it's blood? I would say, well, it's certainly possible. I mean, there could be other things that migrate to a similar position. Like in a lot of these studies, I would like to see non-consecrated wafers just to see, well, if you load that as a control, where does it go? But it's certainly a reasonable experiment. Does it really show that it's truly blood? I would say again, possibly, but it certainly wouldn't tell that it's human blood. So we'll move on to the next one. This is called electrophoretic trace. It's a similar kind of idea that it is based on migration where it goes, except for here you give it a shot of juice, you give it a shot of current. So this is normal blood here. It'll separate into various fractions. This is the main one, albumin, a blood protein, and these are various globulins at these migration places. So here's the result from the Lanciano paper. This is the actual sample. It doesn't show blood, but says that it superimposes right on top of it. And it's a pretty reasonable result. I mean, you have a large peak here that would be analogous to where the albumins go, and then globulins that run here. So does this show that it's blood? I would say, probably, but it's a relatively, it's not as specific as other tests that are now available. And certainly it doesn't show that it's human blood, but I would give it a step up from the last one. So in Dr. Serafini's book, he says that it's qualitatively unquestionable that the miracle of Lanciano contains true human blood. And I think that unquestionable is a very strong word. So I would have a different opinion about this. So we're going to look at the experiments that try to dial in what particular species that might be. So for this, you would go to immunological test. And let me just put out a couple of words here. We've talked about antigen when we talked about A and B. Where that word comes from, what anti means is against, and gen is like genesis, to produce or to cause. So normally an antigen would go in the body, it would stimulate the immune system to respond. And one way it responds is to make antibodies, which would bind to the antigen. For our discussion, what an antigen is going to be is just something that an antibody recognizes. So we're going to have an antigen antibody complex. So for this type of study, this is an older kind of technique. It's a precipitin test is what it's called. But the basic premise for all of these immunological tests haven't changed. It's antigen antibody recognition. The way we can do them is a little more sophisticated, but at the basic level, what goes on is that antigen and antibody recognize each other. So in this test, you put antibodies here down at the bottom of the tube, and then you put antigens at the top, and where these meet and recognize each other, you'll get this kind of complex formed. And in real life, it looks something like this band here. So we're looking for where these antigens and antibodies come together. And these are the results that he published in this study. And let me start off by just pointing this out right here. So this thing that you see all the way across, this isn't really that band. This is just the fill line of where the liquid stops, because that's all the way the same across all of these tubes. So we don't really want to look at that. We want to look below that fill line, and that's where the action is going to be. That's where the band is going to be. So I'm going to start with this third tube from the left. This is the control. This is going to be the gold standard. So I've labeled this in gold. Here he's taken an antibody to human blood and he's mixed that with human blood serum. And if you look again below that fill line, you see that you get a positive band. So this would be as good as it's going to get. This is the gold standard. So I'm now going to hop over to the very far right. So this is a negative control. He takes this same anti-human antibody against human blood, and instead of blood, he just adds saline or salt water, which there's no reactivity. You don't see any reactivity. That's what you expect. It's a good negative control. Next tube over, the same anti-human antibody again, and now he's going to add ox serum. I think the ox may be something in the translation. So the original article is in Italian. This is most likely bovine or cow, I'm guessing, but it translated as ox, so I just left it at that. So here he has antibody against human blood. He mixes it in ox blood, and you get a negative result. This is just to try to show that the antibody is specific for human blood. But from an immunology point of view, what you would want to see is a tube where you took this ox serum and you added in an antibody that in fact recognizes ox blood, just to show that there's really something in there in the serum that's functional on that day. Now, someone can say, well, you're just trying to be kind of picky at this thing. We know this ox serum works because we tested it before in other experiments and it works. From an immunology point of view, you have to show this other control the same time that you do all of the other tubes to show that on that day, this serum truly was functional. It has to be included from an immunology perspective. So because it's not, I'm going to discard this tube. I'm going to go now to these guys right here. So they used actual sample from the miracle for these. So this is blood. I think it was actually a blood rinse and a tissue sample. And now they're going to use a different antibody. This is antibody against rabbit blood. So you can see that there is no band that's formed. There's no reactivity. But again, what we don't have is any validation that this anti-rabbit antibody was really functional that particular day. It's not enough to say, well, we used it before, we use it all the time and it works okay. You have to include it in that same day under those same conditions to verify that it's truly functional. From an immunology standpoint, that's essential. So so something like this, you would use that same antibody, anti-rabbit, but you would add in genuine rabbit blood serum to show that that antibody is in fact capable of recognizing that it's functional. So because that control is not included, I'm also going to discard these two tubes. So I'm going to let these three go and say that, unfortunately, because those other controls aren't included to validate and verify that the time study was done, run in parallel, I'm going to let these three go. So now we're going to go over to this left-hand side. This is where the main action is. This is the anti-human antibody, and this is the blood rinse. You can see you get a band here, reactivity. The tissue section, you can see you get a band of reactivity. So this is a very good result. It's the kind of thing you would hope to find, that it does react. But because this is kind of such an extraordinary thing, I would like to see some additional controls. I've just caused it, for lack of a better term, faux blood and faux tissue, something with like non-consecrated wafer rinse or something like that, just to help assure that this isn't just non-specific sticking that's occurring. Antibodies, they specifically recognize things, but when you work with them, they will also non-specifically recognize things. You have to have those conditions set where you can show that, well, this binding truly is in fact specific. So I would like to see something like this just to help strengthen the results. I would be more comfortable with the true specificity of this. So as far as being qualitatively unquestionable that it contains true human blood, I would say this. I would say that in my own opinion, the nonspecific reactivity of the antibody can't be ruled out. You need to have additional controls included for verification. Now, if you wanted to include all of those other tubes, and let's say those controls were run and everything was fine, that all those tubes passed the mark, none were discarded. I would also point out that cross-reactivity from an immunology standpoint is a major concern. So the question I would have, well, does this antibody preparation exclusively recognize human blood? That's not demonstrated in the study, even if all of those other tubes were included. So let me just talk a minute about how this kind of thing often happens. This is the same kind of thing with the shroud. Well, they say they used anti-human antibodies. Why are you saying that it doesn't really show that it's human? So when you get a vial of something that's an anti-human antibody, so it's you buy it because, well, it reacts with human products. All that anti-human designation refers to is what the antibody was made against. So when it says anti-human, what that means, it means that a human protein was used to generate that antibody. So usually put this in another species and it makes antibodies. So it's anti-human is how it was made. It was made against a human protein, but that doesn't mean that it only reacts with a human proteins. It can react with that same protein in a lot of different species. So this is kind of a misunderstanding. I think a lot of even investigators, scientific investigators, they don't take it past the label and really look into this further. Now, this is a modern day screen capture from an antibody catalog, just a random antibodies, just to kind of show you how this works. So over here on the right, they say known species cross-reactivity. So notice how they word it. They say it's what's known, which means there could be some others that they haven't really tested. But if you look at just one kind of picked out here, you can see this antibody, it reacts against rat, human, mouse, hamster, and chicken molecules. And some of these others you can see up here, they react with numerous types of species. So when you see this anti-human, it only indicates what the antibody was made against, not what it has potential to react with. So these antibody preparations used in this study, so you're talking back in the 70s, these are going to be very crude antibody preparations. It's almost a guarantee that these would react with blood from other species. So as far as claiming that it's demonstrated that it's human blood, I don't think you can make that claim, especially unquestionably demonstrated. I don't think you can make that claim, even if you're willing to include all of those other tubes, which I discarded for other reasons. So this is a common thing that comes up in these type of studies. Same with the shroud. Well, we used an antibody against a human product, but again, all that means is that anti-human designation just means how the antibody was generated. It does not address what it could potentially react with. And there may be species they haven't tested that the antibody could react with. A lot of times from investigators, it doesn't make a difference. If you know that the cells you're using are human, if it also reacts with the mouse or chicken, but it doesn't matter because you know what the origin is. But when you have these unknowns that you're trying to show that what the species designation is, you have to really take into account that cross-reactivity. So I would say that these studies do not take that into account. Also, with the Lanciano miracle, you have to mention the WHO report. So this is a supposed report done by the World Health Organization that verified the physical presence of true flesh and true blood over, I think, 500 tests that were reported to be done. And so Dr. Stacy Trasankos has also written a book called Behold It As I. She's a scientific Catholic writer. She describes the backstory of this in some detail. Dr. Serafini mentions this in his book as well. Along with others, I contacted the World Health Organization to ask about this report. They had zero record of it. in their archives had never heard of it. This was supposedly promoted by the United Nations. I contacted the UN, nothing in the archives. They had never heard of it. Dr. Serafini is the one who went the extra mile and tracked this down. The report was in a vault in a monastery. And what the situation was, it was a cover page that said, Lanciano studies, but the report itself inside was about, I believe, Egyptian mummies, completely unrelated. So he called this in his book a terrible fraud, a terrible hoax, very sad. And a lot of respect and commendation to him for putting this forward. He certainly didn't have to make it public, but I think he's after the truth, which is why he did.
Do you mind if I jump in real quick
Jump
with
in, please, jump.
so I want to echo what you just said about Dr. Serafini and how my impression of his book was that he is after the truth. Um,
Absolutely.
However, I do think that some of the statements in the report, like it's unquestionable that this is human blood, I think that some of that stuff can be traced back to something that he actually says in the introduction. So within the first couple of sentences of the book, this is in the introduction, he says, um, I should immediately declare my conflicts of interest, as is usual nowadays in medical conferences and scientific publications. I am Roman Catholic. I initially thought I could manage writing something aseptic and impartial. I thought that the implied sublime and embarrassing truth would come across to the reader by simply reporting scientific facts without needing comments by the narrator. I couldn't manage this. Like a football supporter in the wrong stadium corner, every time my favorite team scored, I could not help shouting and raising my hands despite being stared at by everyone else around me. So I won't hide my own Catholic point of view, although, my word of honor, this is not a reason for me to censor, attenuate, exaggerate, or otherwise even slightly modify the reported data that are fully verifiable in the annotated bibliographies at the end of each chapter. And to that end, I think that he succeeds. He does do a great job presenting all of the reports in their original, the bibliography is fantastic. But I do think that he gets excited when things look good for his case. And then I think he sometimes oversells it by using words like, unquestionable, when I don't think that that word really applies. That's just kind of my 2 cents.
Yeah, I'm glad you brought that up. A lot of respect for him for pointing that out. That he does kind of have that attachment to it. I mean, I'm a Catholic myself, but scientifically, I try to go into the Dr. Spock Star Trek mode and just try to be completely objective best I can. Yeah. And again, as I mentioned in the start, I mean, I don't have any problem that this kind of stuff could happen. It's not an issue for me. I certainly believe it could, but just trying to look objectively at what's been reported, you see words like unquestionable, et cetera. You have to really look at the strength of the data. Yeah, especially from these are immunological tests. You have to look at them from an immunological background. And that's my previous training. My degree is actually in immunology. So this stuff just kind of jumps out to me when I see this sort of species designation or cross reactivity, human characterization, et cetera.
So
I'm sorry.
no, go ahead. I was just going to ask a follow-up question, but yeah.
Follow up. Yeah. Shoot.
So I'm just wondering, so for the Roman Catholic who is pro Eucharistic miracles,
Mhm.
I can say, okay, everything it's not, we can't prove it's human blood. That doesn't mean it's not human blood. And we can,
You could with today's technology, you could.
okay.
It's certainly possible to do that. I just think it hasn't been demonstrated so far, but it's definitely doable.
Okay, so I was just kind of curious with the Lancia, is there anything that we know about it scientifically that rules out a Eucharistic miracle interpretation, or is that still on the table? It hasn't been proven yet.
I think that could still be on the table. I mean, it occurred so long ago. It's a little difficult, to get all the entire details. You just have to take what's been passed down. But yes, could potentially still be on the table. The hardness of the blood is a tough one as far as analyzing, because most of these you kind of want something that's solubilized, goes into solution, but potentially still on the table. But I think you would have to use a different approach as far as some things you included, different types of antibodies that were used back then.
And
Gotcha.
Dale, just to give you some sense of like time scale. So part of my job is making sure that hospitals around the world send their blood samples to centralized laboratories so that we can do testing to make sure that the medicine that we're working on is doing what we hope that it does. Blood chemistry has an ambient stability. So that's a specific group of tests, blood chemistry versus hematology and stuff like that. We don't refrigerate or freeze those samples. We just ship them ambiently. And the chemistry sample has a 7-day stability, and hematology has a 3-day stability. That is to say, if we receive a blood chemistry sample on the 8th day or a hematology sample on the 4th day, the lab will come back to us and say, hey guys, sorry, we can't test this. If we did, we'd have no confidence that the results are actually accurate. 7 days and 3 days. Compare that to Lanciano, which is hundreds of years. So you could
But
imagine the difficulties.
you could do an approach like with a blood stain, like on the shroud or something like that. You would have to use a different technique than used for fresh blood, but there are tests you could apply for that. Yeah.
Yeah, it's just a lot more difficult.
And yes, it's a long time ago. I mean,
Gotcha.
but I would say, yeah, potentially still on the table.
Okay. All right, cool. So yeah, I'll let you move on. I know your next slide was done. The next one of
I was
Brenner's.
gonna I was gonna talk about something that was done in Tixtula, particularly related to blood, as far as this human characterizations. This was done 2006,
Yep.
so not that long ago. So they did a test for hemoglobin, which is a signature blood molecule. And they conclude the reddish substance corresponds to blood in which there are hemoglobin and DNA of human origin. So I'll just kind of focus on the hemoglobin of human origin. For here, they're called immunochromatography methods. It's a little bit different than the last type of test, but the principle is the same. It's still antigen antibody recognition. So they use these kind of plastic cassettes. A lot of people are probably familiar with this for like home COVID test or a pregnancy test. You load the sample in the bottom here, and then you're looking for a test result line and then a control line just to make sure that the test was functional. If you take this cassette and you open it up, this is what it looks like inside. So over here on the left, this is where you put your sample that you're going to analyze. So this would normally be a drop of human blood that contains hemoglobin, and this is going to hit this pad, and then it's going to travel upstream just by capillary flow, sort of like things just kind of flowing along a river. It's going to travel in this direction. So what's waiting for it first are antibodies against hemoglobin, and these antibodies have been labeled, which is what allows you to see them on the test line. So if hemoglobin is coming down through here, they're going to grab it and they're going to keep going. So here they picked it up. They picked up the hemoglobin and they're moving. They're on their way. You see some antibodies that didn't get hemoglobin. This is intentional. So these antibodies are added in excess. So we're going to have some free-floating antibodies that don't really bind the hemoglobin. So this is going to continue to move downstream. What's waiting for it here is an antibody that's been immobilized or fixed to the plate. This particular antibody is also against hemoglobin. So when these come through and hit the ones that have hemoglobin, they're going to form this kind of sandwich like this. So that's what you're going to see on the test line. These free floaters, they're going to keep going past it because this antibody recognizes hemoglobin. They don't have any, so they're going to migrate past it. What's waiting for them in this next row, this is an antibody that recognizes other antibodies. So it's going to grab these free floaters, and that's the control line. That just ensures that everything moved all the way down like it was supposed to. So I would say from this result, where they report, will we get a positive result? I would say that the binding specificity has not really been demonstrated to satisfaction because these type of tests are heavily optimized. They're optimized for fresh blood. Blood has a particular pH, it has a particular consistency and density, and a lot of these growths or bacterial and fungi products that can be involved in these growths, they've been described as mucilaginous, very, very sticky. So I wonder if you have a non-consecrated wafer solution and you ran it through this kind of test, would you see some sort of non-specific binding? Because yes, these antibodies do recognize hemoglobin, but things can stick to antibodies. You do have non-specific binding. When you work with antibodies, every antibody has its own personality. You have to work those specific conditions out to where that binding is specific. So you don't really know if you could have some non-specific binding that might occur. If you did, it would give you what's known as a false positive. It would look exactly the same. So I would like to see some kind of assurance that non-specific binding really wasn't involved here. I would also say that the claim for human blood species has not been verified. We don't have any evidence that this antibody is specific for hemoglobin. Um, I would say there's probably a good chance that this antibody would recognize blood from quite a few different species. So, cross reactivity for this from an immunology point of view, that would be a concern. I would say that no results are provided showing that it's specific for human hemoglobin only. So, when you make these kind of statements that uh, the results show that it corresponds to blood in which there's hemoglobin of human origin. I think you have to have the data there, the evidence there, to really back up that claim, which I don't think in this case that's really been fully shown. So that would be my opinion of it from an immunology point of view. I don't think that it's not a slam dunk that it's a positive test. Number one, truly for hemoglobin, that some non-specific binding could have occurred, and particularly number two, that the human origin. I don't really think that's been demonstrated in that. So I can pause for that, or I can keep going. Whatever you guys would like.
Kevin, if you have any technical questions on that front, and then
No, not a technical question, just a so this is going to be easy for me to say, Kelly, because I'm not Catholic.
Okay,
Um
sure.
just by this doesn't matter, but I did grow up Catholic. I actually grew up trad, and I was forced into a marriage when I was 22. It's this whole big thing.
Okay.
But that's neither here nor there. But I find it really troubling that um it's almost like an urban legend the way that these stories grow about these Eucharistic miracles. And you'll get these claims written in actual books about how the science is like, uh, unquestionable, right?
Right. Right.
And there's no controls ran, and it's not published in a peer-reviewed journal. And you're like, ooh, Um, so yeah, I just find it really troubling. And I think that if I was somebody who did believe in these Eucharistic miracles, I would be, at my cage, screaming, please do more testing, because I feel like these things could be, especially with today's technology, these questions could be answered. They
Yes.
just
Yes.
haven't been,
Right.
you know?
Right.
So that wasn't a question. I just, I
No,
just
I,
kind of
I think
wanted
it's
to.
a good point. I think for all of these, the goal should be to publish them transparently, publish them in peer-reviewed journals. There's only two. The Lanciano is one, and Austria in 2016, which I'll talk about, that have ever been published in a journal. Scientifically, that should be the end goal
And the
for
Lanciano
all of
one,
these. Yes.
of course. And the Lanciano one wasn't even a peer-reviewed journal. It was Dr. Lannoli's own journal. So still better than Buenos. And the Buenos Aires and the Tixla ones really upset me because there was an investigator, Dr. Ricardo Castellan Gomez, who was in charge of these, and he never had any of his work published outside of like books that he wrote, which, if you ask me, is crazy. Like we had a team, and we can talk about Dr. Ricardo Castellan Gomez. We could talk about the two people who hired him. Because I think that adds a whole nother level of Well, it's even more problematic, I think, when you start looking into who these people are. Um, but again, that's kind of also neither here nor there. So, um,
Yeah.
yeah, let me leave it there.
I have a question, but I think it's best to wait to the end because it's kind of a general thing that can apply
Okay.
to all the cases. So yeah, I'm
Okay.
happy for you to move on to the next case.
Okay. So what's loaded in here is just kind of the stuff we did with non-consecrated wafers, and that's going to launch into some of the other miracles. Is that okay to do it kind of as a background thing for
Yep.
that?
Yep, of course.
Okay. So as I talked about, one of the things that stood out to me, a common theme of these five that are listed here, four of them, 80%, would go this route where there's a discarded host. It was placed in water to dissolve according to church protocol. The reddish substance didn't appear 7 to 10 days later. The one of this list where that didn't happen, Tixtula, I believe that's the correct one, that a nun distributing communion saw reddish growths on a host. So it wasn't submerged in water, but she saw it there in the container, which I believe you can imagine, well, that the bread had gotten moist because of humidity or sweat from somebody touching it, and they'll store that back for a while. So I think you could see you could reach the same result. But most of these very common story: discarded host, too dirty or dusty to consume, placed in water. It appears 7 to 10 days later. So I didn't publish these side by side in the article just because of potential copyright issues. But here's the Circulcal Miracle on the left. And these are non-consecrated wafers that we did 7 to 10 days later in water. You can see in the miracle, if you look, these kind of blackish spots. If you look over here in the non-consecrated, you can see a similar thing. These were identified in our study as fungal spores. So it's a very, very similar appearance. One thing a lot of the miracles described is that the water was not tainted, as though it was kind of a divine sort of miraculous property. An odd property, I think, for blood that's fresh. You would think fresh blood would mix very well with water. In the studies we did with non-consecrated wafers, the water wasn't tainted either. And we took a non-consecrated wafer and put blood on it, let it dry real well for 2 days, then put this in water as expected. It was dissolved very, very well. So the fact that the water is untainted, to me, that wasn't really a divine property. It just seemed kind of strange for blood to behave that way. One of the causative agents that's been implicated in a lot of these miracles is a lot of people have suggested, well, it's a bacteria, Serratia marcescens, which produces a red type of pigment that is very similar to a color of fresh blood. So this is just a streak we did. We obtained that bacteria and cultured it out. You can see that it gives a red colored pigment. Let me just point out, this is one of many different types of bacteria that can produce reddish pigments. Fungi also produce, there's a pretty good list of them that produce reddish pigments, but this is one of the more common ones that's implicated. So we took a non-consecrated wafer and we inoculated it with the bacteria. As you can see, this appearance, it's very similar to what you'll find on the internet for a lot of these miracles. And we looked at this under ultraviolet light because blood does not fluoresce under ultraviolet light. It absorbs it. So this bacterial growth, in that property, it was the same as what you see with blood. It doesn't fluoresce under ultraviolet light. Now, in our case, those reddish growths that we had, they did. They fluoresced orange under ultraviolet light. Again, very different than what blood does. Blood absorbs it, it does not fluoresce. So we saw a orange fluorescence. It's a very easy test you can do for these kind of things. It's just a pocket UV flashlight. So that's one property that could be looked at in these type of miracles just to see, was it non-fluorescent or fluorescent? Well, if it's non-fluorescent, Serratia marcescens could still fit the bill, etc. So from that kind of brings us to the subject of divine DNA and cardiac tissue. So I'll just talk about the DNA side
Kelly,
first.
do you mind if I
Yes.
just ask just a
No.
quick
Yes.
question on the fluorescence before we move?
Sure. Sure.
So with the Shroud of Turin, obviously one of the properties of the Shroud's bloodstains is that there are serum retraction rings which
Yes.
fluoresce. So
Oh, it's a very good point. Yes. They're going to fluoresce a whitish color, very different than what you see here with this. But that's a very good point. It's the serum, the blood itself, the reddish portion, does not. It will absorb the UV. So the reddish portion of blood, which is analogous to this, that does not fluoresce. But those serum rings are absolutely correct. They do, but it's a different appearance. It's more of a whitish type fluorescence than an orange, and you can distinguish those very easily. But yes, that's a
my
very good.
my point is, if it is blood on these hosts, wouldn't we expect the same serum retraction rings to show up? Or
Not necessarily, because if it's just issuing forth whole blood, so you would need some kind of clotting, and then maybe a transfer, some kind of density difference where it's settled out. If it's just issuing whole blood, I would think it would just remain where it wouldn't really settle out that way. Yeah,
okay, cool. Thank you so much.
you bet. Very good question. So for the DNA, these are the three miracles which it's been described, where that's been analyzed. The Buenos Aires, they talk about they got a very low concentration of human DNA, that this is just consistent with contamination and what you might see through handling. But they also found a good amount of DNA from a non-human origin. And again, Dr. Stacy Trasancos talks about this a lot in her Behold It Is I book. She gives a backstory on this. So there was this idea that, well, we had some DNA there, but it appeared to be of non-human origin. And the suggestion for these miracles, it defies detection due to its divine nature. There were some follow-up studies done in the Buenos Aires. I didn't really mention this in the article because there wasn't a lot of detail, but they did a pretty sophisticated technique, single cell sorting using markers for white blood cells. So they sorted those out first, followed by sequencing. They reported for nuclear DNA, there was no human genetic code, but they did find mitochondrial DNA present. Tixtula, there were kind of two runs that were done on this, two sort of tests on it. The first one, it showed the presence of genetic material detected, but no known human coding sequences identified. So when they used probes for human DNA sequences, they didn't see reactivity. The second one, the DNA seemed completely degraded and fragmented, so they didn't really get any results from that look at it. So again, the idea that the genetic material escapes genetic probes. So just as kind of a quick background here, for a human being, the DNA you need to encode, you need nuclear DNA and mitochondrial DNA. The nuclear DNA on the left there, that's 2 copies per cell, 1 copy from mom, 1 copy from dad. About 20,000 to 30,000 total genes are what is needed to encode a human being. Mitochondrial DNA, the mitochondria is just an organelle inside the cell. It has a much higher copy number. It's about 100 to 10,000 copies per cell, which is why mitochondrial DNA is often used, because you have a lot better chance at getting some of it. But it's a much smaller gene pool, just about 37 total genes. Nuclear DNA inherited from both parents, 1 copy from mom, 1 copy from dad. But mitochondrial DNA is strictly inherited from the mother. So it can be very, very discriminatory in trying to see if two individuals are related. So this idea where there's no human genetic code that has been found, or what I call the triple D, the DNA defies detection. What does that really mean? I mean, the human genome consists of 20,000 to 30,000 genes. Does this mean that Jesus would not have a gene for fingernails, or Jesus would not have a gene for hair, or Jesus would not have a gene for hemoglobin? The Catechism and the faith teaches us Jesus was fully human and fully divine. So we would expect to see some of these human DNA signals present. So in our case, when we thought about, well, they find DNA, but it's of non-human origin, it doesn't react against these human-specific probes. What could account for this finding? So we thought, well, the host is made of wheat. Is there wheat DNA present there? The answer was yes. We found when we did PCR to amplify it up, there is wheat DNA present. This was actually shown a number of years ago by Mike Tilley in Kansas. He works for the USDA there. He showed it using whole wheat bread that wheat DNA survives the baking process. So we found wheat DNA present in the host. We also found non-human bacterial DNA, quite a few of bacterial DNA signals present. And our main signal for us, in our case, was fungal DNA. And we were able to narrow it down. Most likely candidate is Epicoccum nigrum. It produces these reddish pigments and they do fluoresce orange under UV. So we think that's most likely what we're seeing in our particular case here. So this is just an alternative explanation as to why you could have a DNA present that doesn't react with human-specific probes. In Lignaika, Poland in 2013. Yes.
Sorry to interrupt there.
No problem. No problem at all.
So when I had Massimo Paris on the show talking about this notion, because Kevin brought this notion of bacteria or red mold or fungi,
Okay.
and his response was to that is that, well, that's just pure speculation. There is no evidence for it. So I just wanted to clarify. Were you saying there was a study that indicated the presence of this?
No. So in the miracle cases, it's to my knowledge, it's never been looked at and never been considered. In our case, with the non-consecrated wafers, we wanted to demonstrate that you would have non-human DNA present, and these are potential sources that you need to consider in those miracle studies. To my knowledge, there's never seen anything written. Well, we did, probes for these, but it turned out to be negative.
Yeah, Kevin.
Yeah,
Okay.
and so like if Massimo's critique of me is something to the effect of Kevin concluded that there definitely was fungal contamination in these DNA cases, then I would agree with Massimo. I didn't actually conclude that, and that's why. But I think it's a real possibility that has never been disconfirmed.
Yes.
And
Yes.
yeah, and in Kelly's paper, there's a demonstration that this is a reasonable explanation for everything that happened. It's a reasonable explanation for DNA being found, but not human DNA, at least not enough human DNA to replicate and stuff like that.
Yes, we show that. I didn't put it in here, but we show.
Sorry, I skipped ahead. I love your paper so much.
Yeah. Oh.
Let me not spoil anything. Sorry about that.
Okay. So here's one. 2013. Now this says they found both nuclear DNA and mitochondrial DNA present, but the results are kept confidential in order not to sensationalize things. But I can certainly appreciate that part of it. But to analyze this, you need to see something more than that. So they say it's proof of the human nature of the tissue beyond doubt. But so let me just kind of point out with all of this, I've never seen just kind of a simple fundamental question addressed to show that a DNA signal is present that originates from blood. So it's a very straightforward thing to do. I've never seen it. I've seen these kind of things with these other variable like STRs and things like that. But I've never seen this kind of fundamental question. Well, how do you know that the DNA is coming from blood and not just skin? So let me just pop into that for a second. I've written about this in terms of the shroud, but the miracles, I think it really applies because the shroud may be a little tougher to get DNA in good shape from that. But from the miracles, I think you'd have a better chance. So Here we have white blood cells, a particular type called lymphocytes. They have these immune receptors on their surface. Skin cells don't. But if you look at the DNA level, skin cells do have the DNA for these immune receptor genes. They just don't use that DNA. And what makes it different and that gives you the power of discrimination in lymphocytes is part of their normal development. They will rearrange and splice this DNA. They're the only cell types in the body that do this. They rearrange it and splice it so it's a much different version than what you find in skin cells. So this is just a lot more detailed slide that it only makes the point that these gene segments in skin cells, they're there, but they're separated by very large distances. Where in lymphocytes, they rearrange and splice these together so it looks much different. And it's a very straightforward kind of analysis that you can do. And you have two different types of immune receptors you can look at and several different gene segments within those receptors that you could look at. So you have a lot of potential shots here to examine that kind of thing. And again, I've never seen this even mentioned or suggested. A very straightforward demonstration that any kind of DNA study you do, you're always going to have potential for contamination with that. But this type of thing, the rearrangement, it doesn't happen in skin cells. So it's a very definitive type test to show that, well, this signal truly is coming from blood cells. I think that's something that should certainly be considered, but I've never seen it really suggested as far as the DNA part goes. So I could pause there if you want to do some DNA questions, or I can go on to the last part here about the cardiac tissue.
Do you think I could play devil's advocate again for a second?
Sure, of course. Yeah, you bet.
Okay. So I'm putting on my Dr. Ricardo Castellan Gomez hat.
Okay.
So I think that the inability to obtain the DNA profile is a confirmation of its authenticity and its supernatural origin. Paradoxically, what you call bad news, I think is actually a reason to be satisfied. I think that the lack of ability to replicate human DNA is a control variable. Every time that a genetic profile, never mind. So I was paraphrasing from roughly page 189 of Dr. Serafini's book. That's in the chapter that he dedicates to DNA. It's chapter 12, the DNA. Okay. That's how Dr. Castellan Gomez talks about it. So if Dr. Castellan Gomez said to you, well, I think that the lack of ability to replicate human DNA is actually proof that this is divine, is there anything that you could say to that? And I can share my thoughts.
Everyone's entitled to their own opinion. I would say these tests they're typically comparative in nature with these STRs and hypervariable regions, and you have to have something to compare it to. And we don't have a Jesus profile to compare it to. But I say the failure for something to react with that, I don't know if that's necessarily a win. There's no. So I would just have a much different outlook on it.
I agree, and my my main concern here is that if we started considering a lack of evidence to be confirmation that our theory is correct, then it almost seems like there's nothing that we couldn't prove as long as we have a lack of evidence for it. You know what I mean?
And, I would say also, so if you look at these unique sequences, well, what about some that aren't unique, like fingernail or whatever? Look at some of those. Is that there? Or better yet, I would go for the one on the skin versus blood, and that's a definitive proof that shows that a signal truly comes from blood cells. Now, he might argue, well, it's unique, that doesn't happen or whatever, but I think you have to have something to show. Just the fact that it doesn't react, I wouldn't call that proof evidence myself.
Yeah. I very much so agree. And I really worry that Dr. Ricardo Castellan Gomez, when he makes a statement like that, he is letting his excitement at the prospect of this be true, like, cloud his scientific reasoning.
Yeah,
That's my
it can
concern.
happen for sure. Yeah, right, right.
Yeah, continue on.
Okay, so that's going to bring us to the cardiac tissue. This is something that's been demonstrated in a lot of these miracles. Here's just a couple: Buenos Aires and Tixla, Mexico. And I've been a number of specialists that have identified these as heart tissue. I've sent these pictures off to pathologists, cardiologists across the country, internationally, and they say, yeah, it looks like what heart tissue you would expect it to look like. Some of the Lanciano, a few of those figures were kind of questionable for some of them, but I think even the initial investigators kind of questioned those a little bit also. Dr. Stacey Trasankos describes that in her book, but they are cardiologists, so you would think, well, they should know what it looks like. I would like to see maybe some other specialists brought into the mix. A lot of these, so here's one from Tixla. They identify a particular type of cell, eosinophil and neutrophil. These are immune cells that respond in inflammation. So a lot of these identifications, they're just based on shape alone. Well, this looks like an eosinophil or a neutrophil. In a normal tissue sample case, it's not really a problem, but this is a bit of an unknown. So here's one from the Austria study. This is only the second one that's been published. They describe structures that could be eosinophils, but they also say, well, these look like they could be fungal spores also. So some of these sections, I think it would be good if people in other disciplines like mycologists, fungal specialists, and things like that also had a look at them. But where you'd want to take this to really help verify it, you would want to take it past, well, it looks like this particular cell type. So we've identified it. You wouldn't want to use cell-specific markers. So there are markers unique to neutrophils and to eosinophils. So you would use a labeled antibody for that and show that, well, it looks like this cell type. And we can, in fact, demonstrate that it expresses a cell type specific marker. That makes the evidence much, much stronger. And there has been some of this work done in the miracles. So glycophorin A, which is red blood cell specific, that gave a positive result. Desmin and myosin, which are specific for muscle, you would think these would show up as positive, but they were negative. So not really sure what was going on there. But this kind of brings us to what I just call the elephant in the room. And I think that evidence has to be provided that all samples originate from a single unique source. And in particular, there has to be an internal scientific quality control check that's included or demonstrated. Because one question that is always going to come up from someone from the outside looking in, if a scientist is examining these objectively, or just a detective working the case, what's the proof that these really come from a single unique source? And not trying to make any kind of assumptions or accusations that samples might have been put in there, but it's a natural question that is always going to exist. It's an elephant in the room, which I think you just have to discuss it. So the end game is to show that these truly originate from a single source. And so that's where you're trying to go with it. And I think it's important to have some kind of internal quality control check just to provide that assurance and to be more objective and transparent about it. So one way you could show that these come from a single unique source, well, nuclear DNA, like some of the tests that have been done, they look for those unique sequences. Mitochondrial DNA, look for those unique sequences and compare them. And they've run into some problems with the DNA analysis, but that is certainly a route that you could go. I would also toss in the HLA transplantation antigens as a potential for that. So HLA transplantation antigens, I discussed this earlier, but you can look at this at the DNA level, but it has the advantage you can look at this at the protein level. So they they show these very pristine, very textbook tissue sections, they would be ideal for looking at this at the protein level. Before DNA technology really took off, this is how they used to do HLA typing, was through antibodies and panels of antibodies. So it's something that could very easily be done. It would also provide a quality control check that these samples truly do originate from the same source, which they should if they're valid. I think until something like that is implemented, it's always going to be an elephant in the room that people are going to worry about, from the outside looking in, just the way a detective would. Well, what I really know about where these samples went, et cetera, et cetera. They could be sure they are looking at cardiac tissue, but what did we really know about that? I think if you have some kind of internal quality control check, that becomes a moot point, and it's a direction toward the desired end game to show that these truly originate from a single source. And that all of this in the studies, one of the main purpose was to try to put together a suggestion of the standard protocol needs to be in place in the investigation of these things with input from people from a lot of different specificities. Because I think if you did that systematically, a lot of these you could just get off at the first exit. There's some cases where they thought a miracle happened and someone comes in and look, no, that's just fungal growth and it's discarded. It just moves on. But I think something like this has to be in place. It's where you're trying to head. It would only strengthen the validity and verification of the data.
I have a couple of questions on my end.
Yes.
All right, so and slow show. So obviously with the heart tissue, one of the
Mhm.
investigators is someone we know well from shroud studies that I at least rely on, and I think you would, Frederick T. Zugby.
Right. Right. Right.
And I think it would be hard. I think we could, on a balance of probabilities, rule out fraud, at least on his end. Like whatever he was looking at was probably heart tissue. What happened before that, who knows, right? But I do want to ask
Yeah.
oh, go ahead.
So I think the who knows, I think that internal quality control check with markers that are specific to to individuals, they should line up from miracle to miracle to miracle to miracle. It would eliminate that who knows kind of question. So no doubt that Dr. Zugby looks at heart tissue and knows what he's looking at, or Dr. Serafini looks at it and knows what he's looking at. Yes, no doubt. I mean, they published the results. Someone could, if it wasn't, I think they would say, no, that looks like this. That's not it. Now, I mean, when I show those to fungal people and said, could a fungal growth look like this? Well, I don't really know. I mean, hasn't really been done from that angle, but I think it would be good to include their opinion. But it's the who knows. I think that is always the elephant. And that's always it's just going to be a natural question that someone is going to raise with that.
So I just want to ask you a couple.
Can I jump in real quick, Dale?
Yeah, sure.
The-
Then I have a couple questions after, and I'm going to share my screen.
Okay.
But yeah.
Okay, this is related to your question, so that's why I wanted to do it now. So, I know that you said that it was brought to Zugabe, Dr. Zugaba, and therefore it probably was heart tissue. And I think that Kelly's explanation there that hey, we can remove all doubt is great. But I also want to stress that this sample from the 1996 Buenos Aires host was first sent to Dr. Robert Lawrence of Delta Pathology in Stockton, California. And Dr. Lawrence said that the sample was epidermis, the outer skin layer. Then it was sent to Dr. Peter Ellis from the University of Sydney in Australia, who also called it skin cells. Then it was sent to Dr. Thomas Loy at the University of Queensland, again in Australia, who said it was skin cells. Then it went to Dr. John Walker in Sydney again, who believed it could have been muscle tissue. Then it was shown to Professor Linoli from Lanciano, who said that maybe it was heart tissue. And then it went to Zugaba, who said it was definitely heart tissue. I don't think it's quite so clear that we can say, oh, well, Zugabe said so, so it definitely is. I don't think it's quite that clear. That's kind of something that I wanted to state.
So
And
again, if you had those cell specific markers in that to strengthen it more than just an observation, it would you, help in big
Yeah.
regard there.
I
Yeah.
don't generally work on AP studies. And AP for the audience is anatomic pathology. Um, and a big part of in pharma when you're doing AP studies, there's a lot of just kind of like looking at pictures of slides and being like, oh, how does this what does this organ look like it's up to? But on the studies that I work on, which I'm grateful for because AP is like a whole different world, it's a lot of just like what is the value of this test that we did on your blood. And I think that there's a decent amount of, I shouldn't call it subjectivity, but there's a decent amount of like interpretation of a picture when it comes to looking at something on a slide versus just, you know, putting the blood sample into a machine and the machine tells you what the value of something is.
Yes, I would agree. So I think also I mentioned in the paper. I think they should not just show those samples, but mix some others in.
Mm-hmm.
Known cardiac tissue, known skin tissue, known non-consecrated wafer infected with fungus, just quite a few of them. And what's your score? What do you see on these individual slides instead of just the samples themselves? I think you ought to mix it up a little bit. My own personal opinion, that's a better way to do that.
Yeah, and
I
Kevin,
yeah,
one possibility, all maybe these scientists were looking at different samples, like
it was the
compared to
same
what.
sample.
Well,
Uh,
that's what's claimed. How do we know there wasn't a swap out at some point? Like that could be an option to consider, right? Like so maybe one scientist was looking at skin cells, and another was looking at, and then Zuckerby was at heart tissue. So that's a possibility.
yeah, I
Doctor,
don't really know. But again, I think if you have that built-in quality control check, it takes you toward the end game you want. It helps control that'll become a moot point.
Gotcha.
Yeah.
So,
and Dale, to your point, I mean, yeah, I certainly can't say that that's impossible, but Dr. Ricardo Castellan Gomez himself said that he prepared one slide and then he sent that one slide to all these scientists. So, but again, none of this was peer reviewed, so I guess anything is possible. I guess
fair.
but
All
it would
right.
seem even worse if there were samples that were appearing and disappearing and stuff.
Okay, so I'm going to share my screen from it's a letter from Zuckerby himself.
Okay.
I just want to, there are a couple things. So I want to ask you on both sides. So one thing that would help the pro-Eucharistic side is so on the helping the skeptic first. So according to Zuckerby, um, the heart was in a certain condition. Like it was, he says the degenerative changes are consistent with a recent myocardial infarction of a few
Infraction,
days
yeah.
duration due to an obstruction of a coronary artery that supplies nutrients and oxygen to that area of the heart muscle. The above type changes suggest that the individual had a heart attack a few days prior to the death due to obstruction of a coronary artery due to whatever. So he's describing certain aspects here, and I think at one point he says he was older, if I'm not misremembering. So the point is, as a shroud skeptic, aren't some of Zuckerby's findings inconsistent with positing Jesus as the source, or do you think it could be consistent still?
Oh, because Jesus. Well, I'm not a physician by any stretch of imagination, but I would imagine some might say, well, his heart was under extreme duress and was stressed, and that could account for this. And Zuckerby was very involved in shroud studies, so he didn't apparently see any contradiction. But I see why you're talking about like a heart attack and a heart attack a few days before. Is that what it said, or
Yeah.
something like that?
Yeah.
Yeah, I don't. The few days before sounds like it may not line up, but maybe the heart was under that much stress. Maybe you'd see similar kind of issues. I'm not really sure about that.
And
Okay.
something I can add
What
to
did you
here.
ask Dr. Serafini about that or?
So he's coming on to do a show in the
Oh,
future.
okay. Okay.
That is something I want to ask him about when.
Yeah, yeah. He would be the one that could probably comment on that the best, I would think.
Yep.
Yeah.
And Kevin, were you about to say something before I get to my next issue?
Yeah, something else I want to say too here is that like if I was somebody who did believe in, I guess specifically the Buenos Aires miracle, I wouldn't get too hung up on um like heart attack a few days before death or like stuff like that. And the reason why I wouldn't is because you simply can't tell all of that from looking at a slide. That's all that Zuckerby did is he just looked at a picture of a slide. That's it.
I
You just
did I did have a similar impression when I read that he seemed to get a lot of information just from
yeah.
that. But I'm not a by no
Okay.
means an expert in the
And
field, but
and neither am I. And I
yeah.
would love to ask an actual cardiologist, but I do work in pharma, and I have worked on studies with AP slides.
Mhm.
And like the AP slides are great for like noticing like very specific things like, oh, look at this, that is there, but then you can't really say how that got there or anything. So saying that, yeah, somebody could have had a heart attack through a couple days before death based on this, that's fine. But there's no way that you can say that this is definitely the case just by looking at a slide. There would need to be further tests done. There was nothing like that. All that Zugabe did was look at the slide itself. And you can't know much just by looking at a slide. I mean, there's things that you can learn, but like if I wouldn't let that stop me from thinking that it was authentic, if that was the only piece of disconfirming evidence. That's kind of my 2 cents.
Gotcha.
Okay. All right. So my next question. Now I'm going to go on the other side. So a pro Eucharistic miracle person might appeal to this letter from Zugabe as evidence against fraud, because one of the things that was surprised, Zugabe was surprised. He says, when I was later told that the heart tissue was kept in tap water for about a month and transferred to sterile distilled water for 3 years, I indicated that it would be impossible to see white blood cells or macrophages in the sample. Moreover, it would be impossible to identify the tissue per se, as there would be no morphological characteristics. So I maybe it's not in this part, but I remember reading that they had to that whatever sample, if it was fried, say they switched out this heart tissue and then Zugabe is examining it, they would have had to have gotten that heart tissue just within like 4 hours or something before it would have been noticeable. Have you ever heard of that aspect? Or yeah, like is it true that he shouldn't be seeing white blood cells and if it
No, they would be gone pretty quickly in water like that. It would surviving something like that'd be pretty extraordinary.
okay. So do you what's your take on if there is white blood cells, is that
I
true?
think it would have to be either a miraculous explanation or an alternative one.
Kevin?
It's kind of all
Yeah.
all
So
I'll say with that.
yeah,
Uh,
I think it's incredibly surprising. Let me put it this way: if indeed there were white blood cells there, I would find that incredibly surprising,
yes, it's
miraculous, even.
yes, exactly.
Now, now I will say this: let's say that you're playing poker with your friends, and your friend gets a straight flush, and then another straight flush, and then another straight flush. And you're like, hey man, I think you might be cheating. And he's like, no man, like it's wild that this happened, but hey, like it's what happened. Like, so that must be the case. Um, I would start to be suspicious that actually your assumption that there was no cheating is just that's simply what's wrong, instead of wow, this really improbable thing did happen. So we could look at our assumption and say, oh, we thought it was a white blood cell, but actually it's probably not. Or we could say, wow, it really is a white blood cell. And that's so super surprising. I think, in light of the evidence, it's more than likely not a white blood cell, is what he was looking at. Um, but again, without further testing, all we're doing is looking at a slide and being like, that looks like a white blood cell to me. Okay, we didn't do any testing, so we don't know if it's a white blood cell or not, but it looks like one. Cool. And the fact that there's something in there that looks like a white blood cell doesn't mean that there definitely is a white blood cell. If there is a white blood cell, I find that incredibly surprising. Um, but I don't think that I'm convinced that there is indeed a white blood cell, if that makes sense.
Gotcha. Yeah. All right. Cool. So, yeah, I guess just to kind of summarize with both of you guys. So, okay, based on the scientific evidence only, what we've been looking at today, Kelly, I just want to ask you overall, are we able to rule out any Eucharistic miracles as not being actual miracles, or are we agnostic on all of them? Yeah. Where do we
I would
sit?
say, so based on the evidence in trying to, well, does this scientifically, are you convinced that this occurred? In my own opinion, for the ones that had been reported, no, maybe they are, but I think you would have to bring more to the table in terms of testing to really demonstrate it. So I would leave the possibility open. But I would say as it stands now, there needs to be further evidence for that.
Gotcha. And I'm just kind of curious, in your studies of the Eucharistic miracle claims, going beyond the scientific data, the data, your expertise,
Mhm.
because Kevin has presented historical issues or, other type of considerations. Have you looked into the other considerations, and do you think those are determinative, in any way as to whether it's authentic or not true, or yeah, what do
So
you?
what type of other considerations again? I'm sorry.
So,
I can list a few, I
yeah,
guess.
go ahead, Kevin.
Yeah, so just a few. So for instance, regarding the Lanciano miracle,
Mhm.
there's a huge issue with the historicity of the actual event. Um,
Oh, I see. I see.
yeah.
So I just for that, I just kind of tried to take it at face value and look at it. What does the science say? So I didn't really. I think there are some kind of gaps, I guess, or whatever you want to call it for that. But I didn't really uh, address that. It would probably be a little bit of an issue for me, but I would my own belief, I would kind of, okay, I'll accept that, but let me just see what the science says and sort of make my determination there. But that's something that's certainly part of the story. Yes, absolutely.
Gotcha. Perfect. Okay. So yeah, I think we're kind of in an agnostic status, basically, is where we're at. But yeah, Kevin, do you have any final questions for Kelly at all?
I do. I have one that I've been dying to ask.
Yeah, okay,
Um,
great.
so I loved your paper, which I have lovingly titled like Making Eucharistic Miracles at Home. Um,
Okay.
uh, but something that I read the whole paper, and I have to admit that, um, I like to talk big game about my scientific knowledge, but at the end of the day, I am just some kid with an engineering degree who works in pharma on the
Okay.
operations side of things, frankly. So I'm not even
Okay.
that. So it's possible that you addressed this in the paper and it just kind of went over my head. But um, did you do any blood typing of the unconsecrated miracle hosts? And if you did you find A and B antigens or whatever?
Yeah, very good question. I did not, because it's a little more involved than what we were doing. So this is basically was a high school summer project, and I didn't think it. I thought there were a lot of other points beside that, which is why I kind of split the AB off
Mhm.
into a separate paper. And I would want to do those the right way with all four types of blood to put it onto the unconsecrated host. And kind of, for lack of a better term, there's a lot of red tape getting different types of blood and things like that to be able to use it. So I would want to have those other types and do that as the controls for that, which is just kind of beyond the scope of it. So we did not do any typing type studies. That's a very, very good question.
Um, Thank you. And if you ever need blood type O, hit me up. I will
Oh,
gladly
are you?
donate
Oh,
some blood for.
okay.
No, but so, and the only reason I asked is because like I thought that the DNA stuff was so cool. The fact that you were able to show like, hey, look, there is DNA present. Like the bread DNA survives the baking process. And
Yeah.
look, that might explain how we found DNA, but we couldn't replicate human DNA. Like that's perfectly consistent here. I just thought that like the knockout punch would have been like, and look, we found A and B antigens being expressed, but it must have been expressed by bacteria because these are unconsecrated. Like that would have been like even cooler. But I thought that the paper
Gotcha.
was super, super cool.
Oh,
That was
thank
the
you. I appreciate that.
yeah, that was the only thing that I was reading. I was like, oh, I wonder if we're gonna get there. That makes sense though. I know it's tough. So I'm only blood type O, but if you ever need blood type O, hit me up and I will ship you some.
Okay. Okay.
Awesome. So yeah, Kelly, just before we close out, I want to turn it to you. Is there anything that we haven't asked you about or that you feel is really important regarding the Eucharistic miracles that you want to let the audience know about?
So just again, just to kind of reiterate, the whole purpose of this was not to try to, hey, we're going to blow these out of the water, et cetera, et cetera. It's just to kind of try to objectively investigate them. And most importantly, for things that are done in the future, to try to have a systematic protocol in place. I think that would be something that would be very important
Yeah.
for this. And I kind of provide a suggestion for one, a basic framework that you could like a working model or something like that. But I think that should really be in place, and to have the end goal of all of this for any future investigations that are they're done, the end goal to publish in a peer-reviewed journal, normal scientific context to make these more transparent. I don't think science by press release is how you want to go with this kind of thing, because it just gets so sensationalized throughout social media and books and everything like that, and needs to go through a proper kind of channel.
What one thing I will ask about because I was working with Dr. Thomas McAvoy, who's
Okay.
involved in shroud. He's published in Applied Optics on the shroud, stuff like that. But in terms of proper peer review, he was showing me, he shared some emails between him and Applied Optics, and like the treatment of some of these secular science peer-reviewed journals towards any religious subject, there is an evident bias there. So I'm wondering
Definitely,
if
yeah.
you,
There's certainly could be, absolutely, yeah. Which I think it's even more imperative. The science has to be above and beyond, and as airtight as you can make it.
yeah,
I mean, even more so, you just kind of have to go up above the extra mile that you normally. I mean, normally you should be very rigorous with science, but for this, you really have to just make sure that you've covered everything. Yes.
gotcha. So I take it on your end, you haven't really experienced because you've been so rigorous in your,
Well, I've tried to be. I've tried to be, and I always try to stay very objective about it.
yeah,
Try not to conclude beyond what the data shows.
gotcha.
Yeah, because yeah,
Cool. All right, awesome. Well, thank you so much, Kelly, for
you're
coming on
most welcome.
and doing this. I really appreciate it. And Kevin, as a great co-host, joining me and raising some great questions and stuff like that. So
And if I may just end on, the last thing that I want to say is that I think that if there were more Catholics like Dr. Kelly, I think that the Catholic Church would be in a better spot. I think that Catholicism would be like a paragon of scientific progress. And so I just want to thank Dr. Kelly for putting out the
You're
papers that
welcome.
you have, for being as rigorous as you are. And for doing so in a way that I get to read it and benefit from it.
Well, thank you. Very
So
kind. I appreciate that. Very, very
thank
kind.
you.
awesome. So yeah, hopefully in March we will be having Dr. Franco Serafini, the
Okay.
immunologist, coming on the show along with Massimo Paris to do a discussion with Kevin. And Kelly was invited. He can show up if he wishes, but yeah, we're going to be having a discussion and going over the evidence, but yeah, with the actual cardiologist on the pro Eucharist side. So
Yes. Sure. Sure.
tune in for that, and otherwise have a great week, everybody.
Okay. Thank you. Thanks again. Thanks so much.
Thanks all.
Bye bye.